I am Cassian Sitaru from the Department of Dermatology at the University of Freiburg, Germany. The research of our laboratory is focused on the pathogenesis of autoimmune skin diseases. One of the autoimmune diseases we are interested in is the epidermolysis bullosa acquisita, or EBA. EBA is an organ-specific autoimmune disease with a well-defined antigen-autoantibody system, and mutually complementary ex vivo and animal models are available for this disease. The blister formation of EBA can be reproduced by passively transferring antibodies against type 7 collagen into mice. Therefore, in this video article, we will present the induction and analysis of skin blistering in mice by the passive transfer of collagen 7 specific antibodies.
Hi, I'm Kinga Csorba from Cassian Sitaru's group in the Department of Dermatology at the University Hospital of Freiburg. In this video, we are going to demonstrate you how the experimental mouse model of EBA is induced by the passive transfer of pathogenic autoantibodies. We use this procedure when examining the possible pathomechanisms in autoimmune skin disorders or when searching for pathogenic epitopes. When establishing the passive transfer model for EBA, a few additional steps must be performed prior to autoantibody injection into wild-type biopsy mice. As a first step, white rabbits are immunized subcutaneously with the purified recombinant antigen. Total IgG from rabbit serum is isolated by affinity column chromatography and subsequently injected into wild-type mice.